Hi, my name is Cecília Magalhães. I am a PhD student in the Institute of Biomedical Science at University of São Paulo, and I'm going to talk about the BMP signaling and the extracellular matrix evolution during early eye development in cheek ink. So the eye development begins with the interaction between a specific portion of the cephalic ectoderm and the optical vesicle that evaginates from the neural tube. So after these interactions, the cells in the preplacoidal ectoderm undergo a cell height increase. So here we can see the transition from a cuboidal cells in the ectoderm to a thick cells here forming the lens placoid. So after this thickening process, the lens placoid invaginates and forms the lens vesicle. So previous studies shows that the BMP signaling is crucial for the lens placoid formation, and also that the BMP inhibition in this optic region affects the lens placoid thickening. So our first question is if the BMP signaling is necessary separately in the ectoderm or in the optic vesicle or in both tissues. So secondly, the fibronectin here in the extracellular matrix of the optic region is also important to the lens placoid formation. And it suggests that the extracellular matrix is important for the early lens morphogenesis. So our second question here is if the extracellular matrix evolves during the lens placoid thickening. So to answer our first question, we inhibited the BMP signaling by overexpressing a dominant negative form of the BMP receptor type 1 separately in the optical vesicle and in the preplacoidal ectoderm. And here in this section, stained with phalloidine, we see the expression of this dominant negative form of the BMP receptor here in the optic vesicle in pink. And we can see that the lens placoid is formed. So you can see here the thick cells that are already invaginating. So the inhibition in the optic vesicle of the BMP did not affect the placoid formation. However, when we did the overexpression of this negative form in the preplacoidal ectoderm, we saw that the lens placoid failed development. So we can see that the cells here are still cuboidal, while in the control eye we have the lens placoid formation. So the lens placoid failed development after BMP inhibition in the preplacoidal ectoderm. Our second question, we analyzed the fibronectin and the laminin immunostaining pattern in 3D confocal images. So here are images of the apical view. So we are always looking at the apical surface of the eye, and we have the eye at different stages of development. In the right, there is an orthogonal view of those images. At the preplacoidal stages, where cells are still cuboidal, both fibronectin and laminin are organized in a fibrillar and punctate pattern. However, during placoidal thickening and invagination, this punctate pattern is restricted to the placoidal region. So it's restricted to the region where we have columnar cells. And also outside of these columnar cells in the extraplacoidal region, this extracellular matrix becomes very fibrillar in both proteins and also in those both stages. So lastly, we know now that the extracellular matrix evolves during lens placoid thickening, and also that the BMP is important for this process. So what is the relation between those two factors? To answer this question, we overexpress the truncated BMP receptor in the ectoderm. So after the BMP inhibition in the ectoderm, we have no lens placoid formation. So we can see here that the cells are still cuboidal. And also when we look at the laminin pattern after the BMP inhibition, we can see a fibrillar pattern that in the control eye is restricted to regions outside of the lens placoid. So we can see that the BMP inhibition in the ectoderm changes the extracellular matrix organization. So to conclude this study, we saw that the optical extracellular matrix organization evolves during the lens placoid morphogenesis, and also that the BMP inhibition in the preplacoidal ectoderm changes this extracellular matrix organization and also affects the placoid formation. And lastly, that the BMP inhibition in the optic vesicle does not affect the placoid formation. So thank you very much. I'll be happy to answer any questions, and please feel free to contact me later.